guys, we are going to go through another chapter. We have new material for this final stretch of the class. We are in chapter 9, starting with molecular biology, DNA, how does it structured, how do we go through central dogma. All of that is covered in this chapter, in chapter 9. So let's get started. When we talk about molecular biology, we're talking about those smaller structures like DNA, the stuff inside of the cell. We're going to go through that. So we will go into cloning at the very end when we talk about Dahlia the sheep, and she was the first clone animal. I saw some recent clones in the news lately when it was talking about, I think it was a black-footed ferret is what they have recently cloned because it's endangered, and so they clone a new one. But we'll talk about clones in a little bit. Let's focus on this first section. That's what I want to focus on right now. So in the first section, we are going to have just a structure. So to begin with, story time, we didn't even know the structure of the DNA until the 1950s. We had done some experiments. If you're in med micro later on, we'll talk about all the different experiments that all the different guys did, all the different scientists did to determine where is the inheritable material. Is it proteins? Which macromolecule is it? Is it proteins on the cell? Is it DNA on the cell? Like what part is it that's important? And so we figured that out. We figured out that it was DNA, but we still didn't know what it looked like. What did DNA look like? So in the 1950s, there were some scientists and grad students, and we have Rosalind Franklin. And so Rosalind, uh-oh, my pen is dead. Rosalind was working on this type of methodology called crystallography, and that's where they are going to use an x-ray and they beam it through a crystal, and that you can look at the pattern that develops on what you're trying to look at later. Like it shows up as this kind of shadow, and now you can see the structure of something. So Rosalind Franklin was using this technique on different things, and she ended up leaving the school where she was at and the lab where she was at and went to another place. I don't think it was the best work environment for her. So she ended up leaving and going someplace else. But later, these other guys came along and they used her research. They built upon her work with crystallography and her discoveries with crystallography. And they were the ones, Watson and Crick and McCarty. Let's see if the pen works now. Yeah, there it goes. Watson and Crick and McCarty were the ones who got the Nobel Peace Prize for this discovery of the structure of DNA. It was thanks to this crystallography work that Rosalind Franklin started that we knew, and here's our shadow, here's our crystallography shadow, that we knew that DNA is a double helix design. We know anything about, what it looked like, and based on the crystallography, we figured out that DNA is a double helix. And so that's going to be important when we talk about how DNA is a double helix and RNA is a single strand, and we compare these two things together. DNA is a double helix design. Rosalind Franklin was doing crystallography, and they built upon her work. And this was, outstanding. This is a huge breakthrough. So it was Nobel Peace Prize worthy, but she did not get it. She had passed away by the time that Nobel Peace Prize was awarded, and they didn't award them posthumously after people die. And she actually passed away from cancer, which is probably due to the fact that she was using x-rays all the time in her crystallography. So a little story time about DNA and figuring out that it is a double helix structure. DNA's single monomer units. We talked about macromolecules, we talked about proteins and carbs, right, and lipids, and our nucleic acids had nucleotides as their monomers. And so those nucleic acids are going to be made out of nucleotides. So there is a phosphate, there is a sugar, and it's tacked to a nitrogenous base. And there will be 4 different choices of nitrogenous bases that we're going to go into. DNA is going to be made out of deoxyribose sugar. In just a little bit, we'll have a picture of the 2 different sugars, DNA and RNA, deoxyribose and ribose. Those are the differences in DNA and RNA nucleotide structure, is that sugar. So our 3 components of our nucleotide for DNA is phosphates dangling off the end, sugar, which is deoxyribose if we're talking about DNA, and then a nitrogenous base. So what are the bases? I believe we mentioned these before, but just in case, this is the time to talk about DNA and all the details about DNA. So DNA's nitrogenous bases, there are four. They group them into two. They are pyrimidines and there are purines. And a pyrimidine is best friends. They are best friends with the purines. Purines are pyrimidines. They're BFFs. They go together everywhere. So when we talk about this double helix structure, we're going to have one nitrogenous base over here, and we're going to have another nitrogenous base over here, and we're going to have a hydrogen bond between the two of them. Actually, several. There will be 2 or 3 options for hydrogen bonds between these guys. So A is always going to bind with T. Adenine A is always binding with thymine T when we are going from DNA to DNA. Adenine and thymine are always together. Cytosine C and guanine G are also always together. Cytosine and guanine, C's and G's, and A's and T's. I remember this with A and T makes the word at, and that helps me remember. A's and T's are always together. C's and G's are always together on DNA. So that is its nucleotide structure. You will be given example of a strand of DNA, and I'm just going to make stuff up. I'm going to say that this is DNA over here, and we are going to make another copy of DNA on the other side. So I'm going to need y'all to know what it is. So I'm completely making this up. Sure. We're going to go with that. So we're going to say that this nucleotide strand here, these are its nitrogenous bases. Let's say that this one is 5, 3, 3, 5. But we need to know what A binds with. What does A bind with? And so we talked about A always binds with T, A and T. So on this side, you'd have a T. This is a T on that side. So what's going to be here is an A, A's and T. Again, A's and T's are always together. This is a C, and C always binds with G, cytosine and guanine. And then G, we got 3 G's, so these are all going to have the same letter. G's are always going to bind with C's. C's and G's are always together. So we are going to have 1, 2, 3 C's. And that's what I'm going to need you to know. If you are given a strand of nucleotides with these kind of nitrogenous bases, what is the complementary strand? What are they going to bind with? You need to know how to do this. So remember, A always goes with T. C always goes with a G when we are talking about DNA. Other helpful things that we need to know about this is that these guys on their structure, they have this blue section over here. This is our sugary phosphate backbone, sugary phosphate. So there is the sugar, deoxyribose, remember, and then there's our phosphate. Where do those phosphates attach? They are going to run. In this example, we can see here is our phosphate up top. Here's our phosphate up top. So this is considered the 5' section, and this is the 3' section. Why is that? If you count up your carbs, 1, 2, 3, 4, 5. That's your 5th carbon, and that's where the phosphate group is. Do I need you to know that and be able to count that out? No, but I do want you to understand how it's set up. Because the other strand, the complementary strand, if this one is running 5' to 3', this side will be 3' to 5'. And so they are considered anti-parallel. Here's my 5 and here's my 3. Then I'm definitely going to have a 5 down here and a 3 here. So these guys are going to go together. They are anti-parallel, 5 and 3 on one side and 3 and 5 on the other side. And that's what anti-parallel means. So understand what's a nucleotide. A nucleotide is going to have phosphate, the sugar, the deoxyribose for DNA, and then a nitrogenous base. And those are our 3 components. Which nitrogenous base is attached to which ones? A's and T's, C's and G's. Always, always. And understand, too, that these guys are running anti-parallel in this double helix structure. Where here are our little bonds, and to me, it looks like like a zipper that you have twisted to make this double helix structure. So that is how it is. Here's our sugary backbone. Sugary backbone. And here's our nitrogenous bases. Here's our T and our A. Here's our G and our C. And here's the hydrogen bonds that keep DNA zipped up. Later on, we're going to talk about transcription and translation, and transcription where we break those bonds because we need to make a copy of DNA. We need to express a gene. We will break those bonds. So that is what keeps the double helix zipped, is the hydrogen bonds between those nucleotides. So that is DNA structure a little bit. Let's talk about RNA structure. For RNA, it sounds just like DNA, except it's got a different... Sorry, guys, that Betty is finding gremlins in the pillows next to me. For RNA, it is going to be ribose instead of deoxyribose. I keep telling y'all these names, but what does that structure even mean? Why is it called deoxyribose and ribose? Let's look at the structure of that sugar. So ribose, here we have this one and this one on these carbons, right? We said this was one carbon, here's the second carbon, third carbon, fourth carbon, fifth carbon. On the second and third carbons, look at these guys. We have these hydroxyl groups that are dangling off the edge there for ribose. So ribose has both hydroxyl groups. Deoxyribose here is one hydroxyl group, but over here we're missing it. There's no oxygen over here, so hence the deoxy, it's missing the oxygen on that little hydroxyl group dangling there. It's just a hydrogen. No oxygen. So that is the difference. The sugars are the difference in the nucleotide structure. On the nucleotide structure for deoxyribose and ribose. Otherwise, other things will stay the same as far as phosphate groups. Phosphate will tack on to this sugary backbone, and then we will have a nitrogenous group. We will have a nitrogenous group where these guys are going to attach. There are different types of RNA. You will see them and they will be abbreviated. Messenger is going to be our first part of central dogma. Central dogma we've mentioned over and over again, where we go from DNA to RNA, deoxyribose to ribonucleic acid to amino acids, are your building blocks for proteins. Messenger RNA is that first RNA that we are going to build coming off of DNA. So messenger RNA, we'll get to this in transcription and translation. We're just setting the stage here. Messenger RNA, transfer RNA is going to bring in the amino acids on each little piece of tRNA. It's going to have a little amino acid tacked onto it. So that's transfer RNA. And then there is ribosomal RNA. Ribosomal RNA. And that is going to be our building blocks for our ribosomes. And those are little guys that are going to, the ribosomal RNA is going to hang out inside the nucleus, inside that nucleolus, inside the nucleus, and we are going to be able to have our building blocks for ribosomes. And ribosomes are, you're going to see, are central to central dogma. We cannot create amino acids without ribosomes. We need them. We need them. Everybody needs them. Eukaryotes need them. Prokaryotes need them. Everybody needs ribosomes. And so ribosomal RNA helps us build those things. So there are multiple types of RNA. We're going to go through them, specifically messenger and transfer is what we're going to talk about in transcription and translation later. But right now, just understand DNA and RNA structure. What are those nucleotides made out of? The phosphate, and then the sugar, deoxyribose or ribose, and then the nitrogenous base, adenine, guanine, cytosine. How these guys are arranged in the cell on nitrogenous bases. Let's just talk about that one more time. We have this pretty picture of pyrimidines and purines for DNA. This is DNA. For RNA, we are going to not use thymine. RNA is going to take that guy out, and we're going to have uracil. We have uracil, U for uracil. So RNA is just going to swap that out. But all the other pyrimidines and purines would be the same. We're still going to have cytosine that's going to bind with a guanine. We're still going to have adenine, but instead of adenine binding with thymine on RNA, it switches and it binds with a nitrogenous base called uracil. So if I was trying to go, I was going to be sneaky, and I said, let's make DNA in our complementary RNA strand, like you'll have to do on your table in the plasmid project. We have, your non-template, then you have your template, and you're going to use your template, and I'm going to create a make-believe template. I'm going to pretend that we have, sure, that's our DNA template. We have these guys here. So we got 2 codons, and we're going to chunk them. And when we go from DNA to RNA, what is our complementary base pair? C's always bind with what? What do C's always go with? Always, always, no matter if it's DNA or RNA, C's always bind with G's, cytosines and guanines. So we're going to have a G here. T's are always going to bind with adenine. Adenine is always there. Here's a tricky one though. A's, if we were going from DNA to DNA, we'd say T, A and T. But this time we're going from DNA to RNA. So instead of T, we're going to sub in uracil. We're going to sub in U. So here's our U. And now we're going to shift on down to the next codon. G is always, again, buddied up with C. C's and G's all the time, no matter if it's DNA or RNA, C's and G's. And then here's a C, so here's a G. And then here's an A, and we are looking, again, remember, this is RNA, so we're not using T, A and U, A and U, A and U. Just in case you ever see that on the test or anything like that, you need to know, and we'll definitely use it on the plasmid project, you need to be able to go from DNA to DNA. What's the complementary template and non-template? And then from you're actually given that on the table because you're going to go down your strand. And then on that table, as we move from our template RNA, template DNA, sorry, we are going to then write down our template, our RNA. So then we'll have chunks of three RNAs we're going to write down in threes, and our little things go down. And then we're going to use that RNA to figure out what amino acid it builds. So we're going to get into that later, but right now I just want you to understand going from DNA to DNA, or are we going from DNA to RNA? And which nitrogenous base binds with which one? So that is what the goal is, one of the goals out of this section. Understand how it's composed and who binds with who is a big thing. Other comparing things. If we're going to compare where do we store the genome? Eukaryotes and prokaryotes both have DNA. Everybody's got DNA unless we're talking about viruses, and viruses' genome may be RNA instead. But we're not talking about viruses, we're talking about prokaryotes and eukaryotes, and where do they keep their DNA? So with eukaryotes, we mentioned this before previously, but let's just bring it back and refresh. Our DNA, me and you, we're eukaryotes, and our DNA is housed inside of the nucleus. Remember, it's got a nuclear membrane. Here's that nucleolus that's going to have our RNA, our ribosomal RNA inside there. Our DNA is in the nucleus. So here are all our DNA in here. Our guys are going to be arranged in chromosomes, just like prokaryotes. Ours are going to be linear chromosomes. Prokaryotes have DNA. It's going to be arranged in the chromosome. It's going to hang out in a different place, though. Instead of being packaged in a membrane like our eukaryotic cells are, these prokaryotes are going to have their nucleoid, their DNA in an area called a nucleoid. There's no membrane there that separates this section from that section that divides out the nucleus. There's no nucleus. It's just an area inside the prokaryotic cell called a nucleoid. And that's where the DNA hangs out. Another comparison, prokaryotes and eukaryotes, nucleus versus nucleoid, linear chromosomes versus circular chromosomes. So on your plasmid project with our prokaryotes are going to have very big, gigantic circles of chromosomes. So they've got this one huge circle. It's massive and it's all folded in on itself. And it's going to be wrapped around proteins that we're going to talk about in just a second. But this big circle has like their core genome or all the basic stuff, you know, how to metabolize their food, how to reproduce, all of that is in this core plasmid and core circular chromosome. But then they may have other additional genes in other things called plasmids, hence the name for the plasmid project. So with the plasmids, these guys are much smaller circles. If we were doing the plasmid projects, there's our eukaryotic gene, but we would create something that is going to be circular shape. When we finish this project, we are going to have something, you can't see all these things, but we're going to have a really big, you kind of see it, a really big circle at the end that's going to include our insulin gene and the prokaryotic plasmid. So plasmids are circular. They're just smaller circles compared to the really big circle that's got all their really good stuff. The plasmids will have all sorts of cool genes that aren't really essential, but they're really great ones. Like maybe they may pass back and forth antibiotic resistance among each other, and they put that in a plasmid. We just looked at E. coli in the plasmid project, and we could use E. coli's plasmid to create insulin and use and harvest their plasmids to make insulin for us. And then E. coli is making insulin for us, and we don't have to use pig pancreases anymore like we used to before we figured out this method. So this is really cool to be able to modify the genes with biotech that we're going to talk about later to create these little circular chromosomes with the genes in them that we want to be in them. So plasmids are circular. Prokaryotes have circular chromosomes, while we are going to have linear chromosomes for our eukaryotes. When we package up the genome, whether it's a prokaryote or eukaryote, it does not matter, but we are going to have, again, DNA is a double helix. It is going to be wrapped around proteins. So if we're talking about macromolecules, nucleic acid is the DNA, and it's going to be wrapped around another macromolecule, a protein. So that is these guys. The DNA wraps around the protein like it's like a tennis ball. And so when we have them all together, it is a nucleosome, and it coils and coils and coils really tight together, because I think I've mentioned this before in a previous video, but DNA, if we put all of our chromosomes, so we have 23 pairs, right? If we put all the DNA in our chromosomes from one cell, we strung it up together, one line, and then the next line, and the next chromosome, we put all of those 23, 46 total chromosomes together in a line, it's 6 feet long. It's 6 feet long. So how do we put 6 feet of information into this teeny tiny nucleus that you can't see with the naked eye? You've got to wrap it up. So you wrap it up around histones, and then we coil it real tight together. And that is going to be our chromatin, hence the name for chromosomes. So we condense, here's chromatin, and then here is one chromosome right here. And then we talked about the S phase when we talk about mitosis and we replicate DNA. When we replicate DNA, it was the S phase of interphase before we got to mitosis. And so this is going to be its sister chromosome right here. So these were sister chromatids that we've talked about before. Sister chromatids. And just one of these, just this guy, is a chromosome, and just this one is a chromosome. But they are carbon copies of each other, hence they are called sisters. So these guys, again, 6 feet of information packaged up into a teeny tiny nucleus. DNA is wrapped around real tight around these histone proteins, and they can pack it together in chromatin, and all of it together is a chromosome. If you're really curious, there is a video on DNA packaging that goes into a whole lot more detail than even I ask of my 1107 of my major's bio. But if you're curious, that video explains it a little bit more about how it's all packaged up into this teeny tiny system of a chromosome. All right, so that's a lot of information. This video was a lot longer than I was looking for, but I do want to, get those basics down, because we're going to build on this basic knowledge of what is the structure of DNA, what is the structure of RNA later. So next video, we will go into DNA replication.